Hi, I'm Andrea Macias, and this is a presentation on purifying beta-galactosidase. So first off, what is beta-gal? It is an enzyme that catalyzes the hydrolysis of beta-galactosides into monosaccharides. These monosaccharides are glucose and galactose. It has 3,075 base pairs and a molecular weight of 456 kilodaltons. It breaks the glycosidic bond so that it is able to hydrolyze the beta-gal. It is an essential enzyme in the human body, and a deficiency in it can lead to a Morquio-B syndrome. This is a picture or image of beta-gal. The purpose of this semester-long experiment is to purify the beta-gal in order to analyze its function and mechanisms. To do this, we conducted a series of steps which were to amplify the DNA, ligate and transform, protein expression, purification, and activity assay. Our first experiment was PCR, which is polymerase chain reaction. The main purpose was to amplify our DNA so that we could use it for future experiments. PCR makes millions of copies of DNA using temperature changes. We use a thermocycling machine which changes the temperature periodically in order to denature the DNA so that it can unwind and eventually synthesize. We use DNA polymerase, Taq polymerase, primers, both forward and reverse, and nucleotides. The three main steps are denaturing, annealing, and extension. For denaturing, when the double-stranded template DNA is heated, double-stranded template DNA is heated to separate into two single strands. Annealing is when the temperature is lowered to enable the DNA primers to attach to the template DNA. And lastly, extending is when the temperature is raised and the new strand of DNA is made by the Taq polymerase. These were our results. So from our results, we could see that our forward primers and reverse primers. We got our forward primer by looking at the first 15 base pairs of the DNA sequence. The reverse primers we got it by looking at the last 15 base pairs and doing a complementary strand starting from the 3' end, and we just basically flipped it. This gel shows us that we correctly amplified our gene of interest because of the column labeled PCR product right here. We can see that a clear band is at the 3 kb mark. The DNA, by using the DNA ladder as reference. Our protein of interest weighs around 3,075 base pairs, which is around the weight of the band. PCR purification was our next step. We did this to purify the DNA that we got from PCR to isolate the DNA of our protein of interest and then clone it. We have to purify it because from the last experiment, additional components like buffers and Taq polymerases remained in the DNA, so we do this to purify it. We added restriction enzymes HIND3 for our forward primer and BAMH1 for the reverse primer. We did this so that in the future we could ligate because it is common after PCR to make compatible ends that allow for ligation, which will be our next experiment. However, PCR purification uses silica membranes that allow DNA to selectively bind when salts that increase solubility of nonpolar substances in water are present. The compounds that don't bind then wash out. Therefore, only our DNA of interest stays. The tris buffer is used to rehydrate the DNA and extract it. So for our results, this is our forward primer and this is our reverse. The ones highlighted in blue are the flanking bases, and the ones in green are the restriction enzyme recognition sites, which is what will be very necessary in the next experiment. This creates sticky ends, which is what we like to see because it makes ligation easier. To determine if our experiment was successful, we can run a DNA gel that separates our compounds by weight. This is just a basic breakdown of what the purification looked like. So our next step is ligation and transformation. The purpose is to make a recombinant plasmid and perform bacterial transformation. Putting a plasmid in a bacterial transformation allows us to make more of our protein. We inserted our BGAL gene into the plasmid using the ligation and the enzyme T4. This was made possible because last experiment we added the restriction enzyme recognition sites and the flanking bases. And they were digested with the same restriction enzyme, so they are able to attach to each other. We introduced the recombinant plasmid into bacteria, which is the E. coli BL21, and the process of transformation. Bacterial transformation allows us to select the bacteria that contains our plasmid by using agar plates with antibiotics. The colonies that were able to form were resistant to the antibiotic treatment and therefore contain our plasmid with our beta-gal protein gene, because the plasmid contains an antibiotic resistance gene introduced by the plasmid. Our results show that we were successfully able to have some viable colonies indicated by the red circles. However, we cannot be 100% sure that our experiment was successful because some plasmids remain without the gene of interest, which is why we have to conduct a test like STS-PAGE on the isolated colonies to make 100% sure that we have our gene of interest. Our colonies grew, so that means that they have the plasmid, but it doesn't mean that they have the gene of interest. And if they hadn't grown, that would have meant that there was a misstep in the transformation process, but not the ligation. But we did end up with viable colonies. STS-PAGE would make sure that the colonies have our gene of interest. The next step is induction. Induction is a process in which specific genes are activated by environmental factors, resulting in the expression of a specific protein such as beta-gal. This process is manipulated in the lab in order to mass produce proteins, which is what we're trying to do. We use the BL21Z3 strain of E. coli because it is commonly used to make copies of plasmids. This is because it is deficient in the cytoplasmic protease. The BL21 is engineered to express the T7 RNA polymerase, which is controlled by the LAC-UV5 operon. This is regulated by IPTG, which is used to induce transcription by the T7 RNA polymerase inside the cell. The T7 will bind to our plasmid, and thus the beta-gal will be expressed. We then conducted lysis so that we could remove cellular debris. We used the freeze-thaw method to weaken the cell membrane and the lysosome to degrade the proteoglycan layer. For our results, after we chose the colony from the plate, we grew it in media and proceeded to induce it with IPTG. This shows the colony which we used for the inoculation. We used the freeze-thaw method so that all the cell components would spill out. By freezing the cells, we formed crystals, and once it thawed, the crystals punctured the membrane and weakened it. The hypotonic buffer also degraded the membrane with the help of the lysosome. This figure shows two bright bands, thus confirming that we have our beta-gal. We took an isolated colony, added IPTG to make various copies using E. coli BL21, and then lysed the cells. We lysed them in order to expose the beta-gal protein so we can move on to our next experiment, which is protein purification. We conducted protein purification because we're trying to get our beta-gal as clean as possible, which is why we conducted these two types of chromatography: anion-based chromatography and size exclusion chromatography. Positively charged proteins flow out because we are using positively charged resin in which only the proteins with a net negative charge bind for the anion-based chromatography, shown right here. We have a pH of 7.5 because at this point, the beta-gal is in its anionic stage, meaning that it will bind to the positively charged resin. The other chromatography was size exclusion. The larger molecules are eluted first. They are bigger than the pores in the non-reactive column and cannot diffuse in the beads. Contrasting is the smaller beads can penetrate the pores depending on their size and thus are eluted last. This one shows the size exclusion chromatography. We did SDS page to confirm that we have, again, the right protein of interest, which is beta-gal. It uses SDS page and polyacrylate gel to separate proteins by size. The mesh-like matrix allows for easier protein separation. The SDS denatures a protein so that it is able to run down. The smaller molecules run down faster and the larger ones slower, so they stay at the top. These are our results. Figure 1 shows us our chromatogram for our anion exchange. And we can see as buffer B goes up, the NaCl concentration also increases. So we end up with around 5 peaks. Figure 2 shows us the SDS page conducted after the anion exchange. The darker bands indicate that we have the proteins we wanted to purify. Figure 3 is the chromatogram for the size exclusion. We have bigger peaks. The later peaks should be smaller proteins. The big peaks are a good indication because our beta-gal is the biggest protein. So the SDS page once again confirms that we have the beta-gal. We can see that we have a decent amount of beta-gal with some contaminants, but the last line shows our beta-gal. The next step is BCA assay. We can conclude that our beta-gal protein was successfully isolated and it is present throughout the experiment because both of these SDS pages show bands at the 114 kilodalton mark. The next step is BCA assay. It is for the purpose of determining the concentration of our purified protein. The bisynchronic acid assay allowed us to determine how much of our protein of interest we have by observing a color change from light green to purple. We use BSA to generate a standard curve to calculate a formula and the unknown protein concentration by plugging in the absorbance of our unknown. The BCA molecule copper complex is what forms the purple color. Two main reactions, reduction of copper 2+ to copper 1+ by the peptide bonds in proteins, and the second one being two BCA molecules chelate into the copper 1+ ion and the two nitrogen atoms in which each molecule reacts with the Cu. These are the two main reactions of the BCA assay. The BCA molecule Cu complex is what forms the purple color. We use a control because the concentration at a certain absorbance level is known, so it is easy to generate a standard curve and compare it to our unknown. We use a spectrophotometer at 562 nanometers. The amount of Cu2+ is proportional to the concentration of the protein in the solution. For our results, Figure 1 shows us the results of our experiments. We can see that the tubes A and B have the light green hue compared to the rest, when the purple color appears and gets opaquer as we continue. We can also see the tubes containing our beta-gal, and by observing the colors and comparing it, it is most closely matches the tube labeled C. We can see in Figure 3 that the absorbance of each tube is corresponding to it in its concentration. For example, tube C, whose color most closely matches that of our beta-gal, has an absorbance of 0.322 and a concentration of 4 milligrams per milliliters. Thus, we expect our protein to have a concentration of 4 milligrams per milliliters. To most accurately determine our concentration, we created a standard curve, shown over here, and that generated this formula, where we plugged in are unknown, and that gave us 4.89 milligrams per milliliter as our concentration. And the final step is an activity assay. This is used to determine whether our beta-gal is functioning properly or not. It checks our protein by analyzing its ability to hydrolyze lactose. If it were degraded or denatured, it would not be able to do so. It would not be able to hydrolyze lactose into galactose and glucose. We use ONPG because the galactose and glucose products are colorless. We use a derivative of lactose, which gives us a color. The ONPG is cleaved, and the resulting products are beta-gal, which is clear, and O-nitrophenol, which appears as yellow. Therefore, we can use this to measure the activity of our protein of interest. We measure the O-nitrophenol's yellow color on a spectrophotometer at 420 nanometers. If the solution turns yellow, then we are able to conclude that the beta-gal is functioning properly because it was able to cleave the ONPG. This is the results. It shows the results of the assay. The clear tube on the left is the blank, and which did not contain any beta-gal, which is why it's clear. On the right is the tube of a yellow with a slight yellow tint. This is the tube that contains O-nitrophenol. This was a product that resulted from the ONPG being cleaved by the beta-gal. This figure shows us the absorbance versus time graph, which is the graph data collected by the spectrophotometer. We use that as our experimental. We can see that our experimental is exponentially increasing and indicative of the yellow tint from the O-nitrophenol. Overall, in order to properly clone our beta-gal protein and analyze it, we first had to conduct PCR to amplify the DNA and make multiple copies. To move on to our next experiment, ligation and bacterial transformation, we had to purify DNA and use restriction enzymes HIND2 and BAMH1 with flanking bases. We conducted ligation and transformation to determine that it was successful, we ran an STS page. We then did inoculation to grow up the colonies to try and express B-gal protein. Later, we conducted 2 chromatography experiments in order to get the cleanest protein as possible. We then conducted STS pages to confirm the presence of beta-gal. We were able to successfully purify and identify our beta-gal. The BCA assay helped us determine the concentration of our protein. We determined we had around 4 milligrams per milliliters of protein, which is a fair amount. The last step was to conduct an activity assay to determine whether our purified protein was functioning properly. We used ONPG and were able to determine that our protein is active. And now we can now move on to one more experiment, which would be another activity assay that would tell us how active our protein is. We don't have to do this, but it would tell us not only if it was active, but how active it was. Overall, our semester-long project was successful because we were able to properly grow up and isolate beta-galactosidase and determine that it was functional so that we could see its function and mechanisms. This is my references, and thank you.